Hello everyone, myself Dr. Gulab Singh, Assistant Professor in Department of Biotechnology, School of Basic and Applied Science, Maharaja Agarsen University, Baddi, Himachal Pradesh. Today we are going to discuss about the BLAST. BLAST is a basic local alignment search tool used for comparing gene and protein sequence with the other sequence present in the biological database. BLAST is a set of sequence comparison algorithm used to search database for optimal local alignment to a query sequence. Query sequence is called the sequence which we want to compare with the sequence present in the database. The sequence which want to compare is known as query sequence, and the sequence present in the database is known as the reference sequence. It breaks the query sequence and the database sequence into fragment and seek matches between them. The BLAST first of all break the query sequence and the database sequence into the fragment, then the fragment are compared and matched between them. The nucleic acid and protein alignment were time consuming, as we are generally use manual method for nucleic acid and protein sequence alignment. It takes very much time, and it's time consuming. Generally alignment were done by using dynamic programming before the development of BLAST. Until the BLAST is developed, the journal alignment is done using the dynamic programming tools. The BLAST is nearly 50 times faster than the dynamic programming methods. Background of the BLAST. Beginning in the 1970s, scientists began to accumulate DNA and protein sequence data at an exponential rate. In fact, researchers currently have nearly 97 billion base sequence and over 93 billion records. Amazingly, this sequence data double every 18 months. Today, one of the most commonly used tools to examine DNA and protein sequence is the basic local alignment search tool, also known as BLAST. BLAST is a computer algorithm that is available for use online at the National Center for Biotechnological Information website and many other sites. Type of BLAST: nucleotide BLAST or BLAST-N. This program gives a query DNA, return the most similar DNA sequence from the DNA database that user specify. In BLAST-N, we use the nucleotide sequence as a query sequence, which may find out the similar sequence. BLAST find out the similar DNA sequence from the database. And in another type, BLAST2, protein BLAST or BLAST-P. In this program, protein sequences BLAST and similar protein sequences find out from the database. Third one is position specific iterative BLAST or PSI BLAST. This program is used to find the distant relative of a protein. To find the similar kind of protein or distantly related protein from the database, we use position specific iterative BLAST. Next is Sixframe Translation protein BLAST or BLAST-X. This program compares the six-frame conceptual translation product of a nucleotide query sequence against a protein sequence in the database. Next one is nucleotide six-frame translation with nucleotide six-frame translation or T-BLAST-X. The purpose of T-BLAST-X is to find very distant relationship between the nucleotide sequences. Next one is protein nucleotide six-frame translation. In this type of BLAST, we use the protein sequence and compare them against the sixth frame of nucleotide sequence. Means the first we break the nucleotide sequence into the six fragment, and then this sequence is compared with the protein sequence. Large number of query sequence, we can use megablast to compare the large number of query sequence. When comparing large number of the input sequence by the command line BLAST, megablast is much faster than running BLAST multiple times. If we want to align the multiple sequence, one way is to align the sequence one by one, which take very much time as compared to when we use megablast. In megablast, multiple sequence can be compared or multiple sequence can be aligned simultaneously. Of these programs, BLAST-N and BLAST-P are the most commonly used because they use direct comparison and do not require translation. However, since protein sequence are better conserved evolutionary than nucleotide sequence, T-BLAST-N, T-BLAST-X and BLAST-X produce more reliable and accurate result when dealing with the coding DNA. As we all know, the protein sequence are conserved evolutionary. If protein sequence change, the function of the protein is changed. That's why the protein is better conserved as compared to the DNA sequence. So if we compare the sequence, protein sequence with the protein sequence, that provide more reliable result as compared to the nucleotide sequence for the evolutionary point of view. Next, BLAST algorithm. The BLAST algorithm is the fast, accurate, and web accessible algorithm. It is relatively faster than other sequence similarity search tool. Complex BLAST algorithm require multiple steps and many parameters. An overview of the BLAST algorithm, a protein to protein search is as follows. First of all, we have to remove low complexity region or sequence repeat in the query sequence. Means the low complexity region of the sequence is removed and the complexity region of the sequence is removed. Then we break the sequence into the small words known as tuples, like as shown in the figure. Suppose a query sequence P, Q, G, E, F, and G is provided, and we want to find the alignment with that sequence. Then first of all, we break the sequence into words, like word 1 is PQG, and word second is QGE, third one is GEF, and the fourth one EFG. However, the sequence provided is PQGEFG. We break them into the word with 3 letters PQG, QGE, and GEF, and fourth one EFG. Similarly, the list the possible matching word. Then we list the possible matching word from the query sequence with the database. Organize the remaining high scoring word into an efficient search tree. Then we organize the efficient high scoring. When we match the sequence with the database, align with the sequence present in the database. First of all, we find the high scoring words. Then repeat the step 3 and 4 for each k letter word in the query sequence. The word we have created in the previous slide is matched with the database. Scan the database sequence for the exact match with the remaining high scoring words. Extend the exact match to the high scoring segment. Then again we extend the exact match to the high scoring segment of the sequence present in the database. List all the HSP in the database whose score is high enough to be considered. Then we make a list for the high scoring sequence which present in the database to be considered. The score is so high to be considered for the alignment. Evaluate the significance of the HSP score. Then we evaluate the score, high sequence score. Then make 2 or more HSP region in the longer alignment. Then we align the 2 region high scoring sequence and make a alignment. Show the gapped Smith-Waterman local alignment of the query and each of the match database sequence. Report every match whose expected score is lower than a threshold E parameter. Every score is reported whose score is lower than the threshold parameter. Next is BLAST input and output format. Input is sequence in the FASTA format or gene bank format. Output, the BLAST output can be delivered in a variety of format. Means the output of the BLAST can be obtained in various format like HTML, plain text, XML formatting. For NCBI web page, the default format of output is HTML. We can take output in different format like HTML, plain text, XML. But the default format for in case of NCBI website, it is HTML. An introduction that tell where the search occurred and what database and query were compared. The output also show that search where is the sequence search and the sequence were compared from the database. Like I shown in this slide, a list of the sequence in the database obtaining segment pair whose score were least likely to occur by chance. Then alignment of the high scoring segment pair showing identical and similar residue. Then a complete list of the parameter setting used for the search like e-score, bit score, identity, similarity search, and the gaps of the sequence also. The e-value, expectation value. The expectation value is a parameter that describes the number of hits one can expect to see by chance when searching a database of a particular size. Means the lower the e-value, higher is the similarity or higher the alignment. It decreases exponentially as the score of the match increases. As the score of the 2 sequence alignment is increased, the value of the E decreases exponentially. Essentially, the E value describes the random background noise. Generally, E represents the random background noise for the alignment during the alignment. In general terms, smaller the E is the more likely the match is significant. The value is smaller. When the value of E is smaller, the match which we found in the database is more significant than the value of E than the sequence which have higher e-value. Default e-value for blast n, blast p, blast x and t blast n is 10. The default parameter set in the NCBI website for e-value is 10 in case of blast n, blast p, blast x and t blast x. T blast n. At this setting, 10 hits where score equal to or better than the defined alignment score S are expected to occur by chance. At the default setting of 10 hits, score equal or better than defined alignment score are expected to occur by chance. The e-value can be increased or decreased to alter the stringency of the search. We can change the value of e-value from 10 to 20 or 10 to 5. It increases the stringency of the search. Increase the e-value when searching with a short query since it likely to be found many times by chance in a given database. When we increase the e-value, the sequence which we find by chance are increasing in the database. Next important parameter is bit score. A bit score is another prominent statistical indicator used in addition to the e-value in a blast output. A bit score measures sequence similarity independent of the query sequence length and database size and is normalized based on the raw pairwise alignment score. Generally, bit score is another parameter which tell about the similarity of the search. It's independent of the length of the sequence and the database amount of the sequence present in the database. This is the website which shows the NCBI home page and different link for the function which we can perform on the NCBI website. Here the red mark circle show the blast link where we can use the blast for sequence alignment. When we click on the blast, when we click on that red circle or blast, this home page is open where different type of blast are shown. The different type of blast like nucleotide blast, blast X, blast T blast N and protein blast. It's dependent upon our requirement which type of blast we want to use. If we want to use a nucleotide sequence against a nucleotide sequence, then nucleotide blast is used. If we want to compare the protein sequence, then protein blast is used. And if we want to compare the protein sequence with the translated nucleotide sequence, then T blast N is used. Similarly, different selection of the blast is depend upon our requirement. When we select a nucleotide blast, this web page is open where the first box is shown for the paste of the sequence, where we can input our sequence in the FASTA format or gene bank format. Then the database is selected. After the database selected, all the similar parameter are remain static. We do not need to change the parameters. Just select the type of blast and we click on the blast button. When we click on the blast button, next web page open which show the similar sequence present in the database, like as shown in the figure. The sequence which show higher similarity are shown in the red color, and the sequence which show different level of similarity shown in different color coding as shown in the frame of the figure. The result out the top segment display the color key and the query based sequence like black, blue, green, pink and the red. The colored bar represent the actual high scoring segment. The position of each bar indicate the region of the query and the high scoring pair. The thin line indicate the two HSP are from the same sequence. The thin line represent the two high similar sequence are from the same sequence. Small vertical line not obtained here indicate breaks. If vertical line are present, that indicate breaks segment which are not connected in the actual alignment of the sequence. What is the function of the blast? Blast can be used for several purposes, including identifying the species, locating domain, stabilizing phylogeny of the given sequence, DNA mapping, comparison of the sequence, etc. First of all, we discuss about the identifying the species. How we can identify the species of the sequence with the blast. With the use of blast, we can possibly correctly identify a species or find homologous sequence. When we have suppose we have a sequence we isolate a microorganism or either a specific type of tissue, then isolate the DNA, sequence that DNA, and we use blast to find the similar sequence from the database. Uh, so the similar sequences, the homologous species, the similar sequence is present in the homologous species. When the homologous sequence find out on the using the BLAST, we can identify that homologous sequence present in particular individual represent the particular species. This can be useful, for example, when we are working with the DNA sequence from an unknown species. So BLAST can be used for the identifying the particular species. Next one is locating domain. When working with the protein sequence, you can input it into BLAST. Suppose we want to identify the specific domain present in the protein sequence, and we want to locate the specific position of that domain to locate known domain with the sequence of the interest. So we again BLAST the protein sequence with the database sequence and find the similar sequence in the database. So the database contain the all the information of the submitted sequence. So we can identify the particular domain in the similar sequence and can assume that similar domain will be present in the query sequence. Stabilizing phylogeny. Using the result received through the BLAST, we can create a phylogeny tree using the BLAST web page. A link is provided on the BLAST website. Using that link, we can create the phylogenetic tree of the query sequence, and that phylogenetic tree, through that phylogenetic tree, we can establish the phylogeny of the particular sequence. Next is DNA mapping. When working with a known species and looking to sequence a gene at a unknown location, BLAST can compare the chromosomal position of the sequence of the interest to relevant sequence in the database. Similar example is here. We have the query sequence compared with the database sequence. If and we find the similar sequence in that particular specific chromosome at specific site. If similar sequence is present in specific chromosome at particular site, means the same sequence is present in the same chromosome at that particular site. So we can map the DNA through the BLAST. Next is compare. The most common function of the BLAST is to compare the gene sequence or protein sequence. When working with the gene, BLAST can locate common gene in two related species and can be used to map annotation from one organism to another. Next, the objective of BLAST. It is one of the most popular program for sequence analysis. Enable a researcher to compare a query sequence with a library or database sequence. Identify library sequence that resemble the query sequence above a certain threshold value. The objective is to find high scoring ungapped segment among related sequences. Alignment of the high scoring segment pair show identical and similar residues of the sequence. Thank you.